This talk is about mutations responsible for resistance to the integrase strand transfer inhibitors, most commonly referred to as Instis. These are my disclosures. 2 chemical steps are required for HIV-1 integration into host chromosomal DNA. 3' end processing, in which integrase cleaves 2 nucleotides from each 3' end of viral DNA, and strand transfer, in which integrase inserts the ends of the viral DNA into the target chromosomal DNA. Each monomeric enzyme has 3 domains: a central catalytic domain that extends from position 51 to 212. It contains the active site and most of the Insti resistance mutations. The N-terminal domain, positions 1 to 50, plays an important role in the formation of the enzyme multimer. The C-terminal domain is involved in DNA binding. Each 3' end reaction is performed by integrase enzyme dimers, while the strand transfer reactions are performed by integrase enzyme tetramers bound to both viral and host target DNA, as shown in this cryo-EM structure. The Instis block the strand transfer reaction by interacting with the bound viral DNA and integrase enzyme in a highly selective manner. This figure shows the chemical structures of the FDA-approved Instis. The chelating motifs that interact with magnesium cofactors in the integrase active site are highlighted with a blue circle. The halobenzyl moieties, which bind to the centralized pharmacophore by a linker group, are circled in red. The Insti resistance mutations interfere with binding of Instis to the pharmacophore formed by the enzyme and the viral and target DNA. This talk will review 3 types of data that inform our understanding of Insti resistance. The first will be an analysis of which integrase mutations are selected in persons receiving each of the Instis. The second will be an analysis of how Insti-associated mutations affect Insti susceptibility. The third is data on the response to treatment in persons receiving the second-generation Insti Dolutegravir. The Stanford database contains integrase sequences from about 30,000 Insti-naive persons and from about 3,000 Insti-experienced persons. Ideally, we would like to have complete integrase sequence data from each patient, but for the second-generation Instis, we often only have the list of drug resistance mutations provided by authors, which could be missing important data. Additionally, there are few data available from persons who receive bictegravir or cabotegravir. The lack of data for bictegravir is likely due to its being rarely used in salvage therapy situations, especially as compared to dolutegravir, and because it is primarily used in upper-income country settings where persons with HIV undergo frequent virological monitoring, resulting in a reduced likelihood of emergent resistance. The Insti query form is similar to that for each of the other drug classes in that it is possible to specify the drugs received by the persons from whom the sequences were obtained and/or the mutations present in the sequences. In this form, we have selected sequences from persons who received dolutegravir as their only Insti without specifying that any particular mutation must be present. This slide summarizes the mutations observed in persons receiving dolutegravir, cabotegravir, raltegravir, and elvitegravir. Dolutegravir selects primarily for signature mutations at 4 positions: G118R, Q148HR and K, N155H, and R263K. The most common accessory mutations include E138K, T97A, and G140A and S. Other less common drug resistance mutations include H51Y, T66I, E92Q, and S147G. Fewer data are available for Dolutegravir. It selects primarily for 3 of the Dolutegravir signature mutations, Q148R, N155H, and R263K. In contrast to Dolutegravir, Q148R and N155H are the most commonly selected drug resistance mutations. Raltegravir and Elvitegravir have overlapping resistance profiles, with mutations at positions 148 and 155 being most common. However, Raltegravir uniquely selects for mutations at position 143 and is more likely to select for G140S and Q148H, while Elvitegravir is more likely to select for mutations at positions 66, 92, and 147. In the few case reports of virological failure with emergent resistance in persons receiving Bictegravir, R263K has been the most commonly reported drug resistance mutation. This slide shows a much more detailed picture of the patterns of drug resistance mutations selected by Dolutegravir in persons who were previously Insti naive before they received the drug. It is from a recent review in which we analyzed sequences from 99 previously Insti naive individuals from 37 publications published through August 2023. Dolutegravir selected Insti-associated drug resistance mutations clustered into 4 largely non-overlapping mutational pathways characterized by amino acid mutations at the 4 signature positions R263K, G118R, Q148HR and K, and N155H. In fact, 82 or about 83% of the 99 virus sequences contain just one of the signature drug resistance mutations, while only 8 viruses contain more than one signature mutation. G118R and R263K were significantly negatively correlated with each other and with Q148 mutations and N155H. This table shows the total number of phenotypic susceptibility results in the Stanford database for each of the Instis. And this figure shows the form for looking up Insti phenotypic susceptibility data. The form shows that 2 mutations are selected, G140A and Q148R. Dolutegravir and the PhenoSense assay are also selected. The figure below shows how the results are presented, with the mutations divided into the categories major, minor, and other. The fold reduction in susceptibility is shown at the far right. The highest fold reductions of 13 and 18 occur when additional mutations are present, E138K and L74M in these 2 examples. The asterisks after the isolates in the first 2 rows and in the 5th row indicate that the complete sequence was not available, and so that additional mutations not reported by the authors may have also been present. Laboratory isolates are site-directed mutants. For these isolates, the mutation list is considered to be complete. This slide summarizes the raw in vitro susceptibility data available for Dolutegravir and Bictegravir. For each pattern, circles indicate the median fold reduction in susceptibility for the combination of mutations shown on the y-axis, while the left and right triangles indicate the interquartile range when there are 3 or more isolates with the same pattern. The x-axis shows the fold reduction in susceptibility on a log2 scale. Viruses that have mixtures of 1/3 or more of their positions were excluded from this figure. In addition, not every drug resistance mutation was used to create a pattern, so there could be some variability as a result of accessory drug resistance mutations that were not included in making the pattern shown on the y-axis. For the same drug resistance mutation pattern, the fold reduction in bictegravir susceptibility is usually but not always slightly lower than for dolutegravir. This slide provides a simplified summary of the dolutegravir susceptibilities shown in the previous slide. R263K alone conferred a median two-fold reduction in dolutegravir susceptibility. With greater or equal to one additional non-signature drug resistance mutation, it conferred a median 3.2-fold reduction in susceptibility. The highest levels of reduced susceptibility of an isolate with R263K was 6.3-fold. G118R alone, or in combination with additional accessory mutations, was associated with a nearly 20-fold median reduction in susceptibility. N155H alone conferred a median 1.4-fold reduction in dolutegravir susceptibility. With one or more additional non-signature drug resistance mutation, it conferred a median 2.0-fold reduction in susceptibility. One isolate with three additional non-signature mutations had a 68-fold reduced susceptibility. Q148H, R, and K alone conferred a median 0.8-fold reduction in dolutegravir susceptibility. With one or more additional drug resistance mutations, they conferred a median 4.1-fold reduction in susceptibility. Several isolates with 148 mutations in combination with mutations at positions 140 and or 138 and or additional accessory mutations had higher levels of reduced dolutegravir susceptibility. Another approach to determining how drug resistance mutations influence drug susceptibility is to create a regression model in which each mutation is an explanatory variable and the fold reduction in susceptibility is the outcome variable. The slide shows which drug resistance mutations are predicted to have the greatest effect on dolutegravir susceptibility. It includes only those 26 drug resistance mutations that occurred in at least 5 isolates in our data set. G118R had the greatest effect. Even though G140S has no effect on its own, it usually occurs in combination with Q148 mutations, and in this setting, it has a significant synergistic effect in reduced susceptibility. Besides the 4 signature mutations, 8 additional drug resistance mutations were significantly associated with reduced dolutegravir susceptibility. We haven't performed the same analysis for bictegravir or cabotegravir because fewer susceptibility data are available for these two instis. The figure on the left summarizes the publicly available phenotypic data for cabotegravir performed using several different methods. Only those patterns for which at least 2 results are shown. The single mutations with the greatest effect on susceptibility were G118R, Q148R and K, and R263K. There are conflicting data between the 2 results on isolates containing G140R, a mutation which has been reported in just 1 patient. Reduced susceptibility is unsurprisingly much higher for those viruses containing 2 or more drug resistance mutations. The 3 figures on the right indicate that for similar mutation patterns, the fold reduced susceptibility for cabotegravir correlated strongly with the fold reduced susceptibility for dolutegravir, especially for viruses containing 2 or greater or equal to 3 drug resistance mutations, with the slope of the lines indicating higher levels of resistance to cabotegravir than to dolutegravir for viruses with the same drug resistance mutations. The Viking trials were the only clinical trials in which participants with baseline Insti mutations received a second-generation Insti. The largest of these was the single-arm open-label Viking 3 trial, in which participants with virological failure and Insti resistance following treatment with a first-generation Insti were treated with an optimized regimen containing dolutegravir 50 milligrams twice daily. The likelihood of virological success was reduced in those participants whose viruses contained a Q148 mutation in combination with one or two additional accessory drug resistance mutations. N155H alone did not appear to be associated with an increased risk of virological failure. There were no patients with baseline R263K or G118R mutations. For every twofold increase in dolutegravir fold change, the odds of achieving a virus load less than 50 copies per mil decreased by 63%. Based on these data, Monogram Biosciences established a lower clinical cutoff threshold of 4-fold and an upper clinical cutoff threshold of 13-fold for dolutegravir. The lower clinical cutoff is the fold reduction in susceptibility at which dolutegravir was predicted to be less active than it would be compared to its activity against a wild-type isolate. The upper clinical cutoff is the fold reduction in susceptibility at which dolutegravir was predicted to have little or no impact on suppressing HIV-1 virus load. Pharmacokinetic data were used to extrapolate these thresholds to bictegravir, 2.5-fold for the lower clinical cutoff and 10-fold for the upper clinical cutoff. The thresholds are presumably lower for bictegravir because its dose cannot be doubled. Although there are few data of cases in which second-generation Instis have been used for patients with Insti-associated drug resistance mutations, there are extensive data demonstrating the efficacy of dolutegravir-containing regimens for treating both ART-naive and ART-experienced Insti-naive patients. This table shows the prevalence of emergent drug resistance mutations in 43 clinical trials encompassing 6 scenarios based on whether patients were ART-naive or experienced, had active virus replication as opposed to being suppressed at baseline, and based on the drugs used in combination with dolutegravir. Among several thousand previously ART-naive patients receiving either dolutegravir plus 2 NRTIs or dolutegravir plus 3TC, there was only one case of emergent resistance among the subset of 140 patients with virological failure who underwent genotypic resistance testing. In contrast, among approximately 1,000 participants in 6 trials with previous virological failure on a first-line NNRTI-containing regimen, the overall prevalence of virological failure with emergent resistance was 1.5%. Among the subset of 113 participants who experienced virological failure and had samples undergoing genotypic resistance testing, 20.4% were found to have Insti-associated drug resistance mutations. Among patients with virologic suppression at baseline, Insti-associated drug resistance mutations only emerged in those receiving dolutegravir monotherapy, but not in the 13 trials in which virologically suppressed patients received a dolutegravir-containing 2 or 3 drug regimen. The data that I reviewed in this presentation are summarized to a large extent in the notes section of the HIV genotypic resistance test interpretation program, and in a very brief format in a PDF handout. The HIVDB website also contains a list of all scores. There are individual mutation penalty scores for nearly all drug resistance mutations, and several penalties that go into effect only when certain drug resistance mutation combinations are present. The total mutation penalty score for a drug is based on adding all of the individual and combination mutation penalty scores. An in-depth description of the HIVDB resistance interpretation program is described in another presentation. All drug resistance mutations that receive a mutation penalty score and some that don't are accompanied by a comment. The complete list of comments for each drug class can be viewed on the website. There is also a table that lists all pre-computed scores for all combinations of drug resistance mutations present in isolates in the Stanford HIV drug resistance database. The table can be sorted by the number of sequences so that the most common drug resistance mutation patterns are shown at the top, or by those drug resistance mutations associated with the highest scores for an Insti. It is very useful for us to check this table to make sure that updates to the mutation penalty scores lead to the results intended for actual virus isolates. Thank you for your attention. If you have any questions or suggestions, don't hesitate to email us at the email shown on this slide.